This is part 2 of the video lecture on the introduction to the lactose utilization system in E. coli. So we left off with Monod obtaining data that showed that his original idea about induction of beta-lactosidase was wrong. It wasn't a preformed enzyme somehow changing shape so that it could now break down lactose. It was rather the enzyme beta-lactosidase only being made when lactose was present, meaning only when the enzyme was needed. And this was a totally new concept in biology, what we now call regulation, gene regulation in particular, and Monod now had a way to study it. Monod was a great collaborator, and his collaborators were geneticists. Two fairly well-known geneticists in France, André Lewolf and Francois Jacob. And these collaborations were critical to the next step in the story. They started looking for mutants, namely mutants that were altered in some way related to this lactose story. And the first mutants they looked for were ones that could not use lactose as a sole carbon energy source. And they found these mutants through a screen. Not a selection, as we've been doing in lab, but a screen. Do you remember the difference? A screen means I have to look at each colony separately and figure out if they're what I want. A lot more work than a selection. Now, the lactose non-utilizing mutants fell into two complementation groups, or two genes. One defined a gene they called LAC-Z, and it, in fact, was the gene that encoded the enzyme beta-lactosidase. Does it make sense that a mutation in the gene for this enzyme would prevent the use of lactose? Well, if you can't break down lactose, you can't eat it. The other mutations defined a complementation group or gene called LAC-Y, and it encodes a membrane-bound, or membrane-localized, permease specific for lactose, called lactose permease. Now, Monod, Lewolf, and Jacob also looked for mutants that would show induction of beta-lactosidase activity in the absence of lactose or any other inducer. And the way they did this is they used the lactose derivative pGal. And I'll remind you from earlier that pGal is a substrate only, not an inducer. So, does it make sense that wild-type E. coli cannot use pGal as a sole carbon energy source, but a mutant that could somehow use it must be making beta-lactosidase all the time? Somehow it had become, quote-unquote, induced, even though there was no inducer present. So this experiment now was actually a selection because wild-type cannot use pGal, right? And geneticists love selections. So what are you going to do? You're going to plate millions or billions of mutagenized E. coli on a plate with pGal as a sole carbon energy source. Now, most of them are going to die because they do not make beta-lactosidase protein. But rare mutants that are making beta-lactosidase in the absence of an inducer will be able to eat the pGal and survive. And it turned out these mutants all fell into one complementation group. And they identified a new gene that they called LAC-I, I for induction. Turned out that this was the first regulatory mutant ever discovered. Now, this gene is not the gene for beta-lactosidase. Right? Rather, it's some gene that's involved in the induction of beta-lactosidase. But it turns out that not only is beta-lactosidase, the product of the LAC-Z gene, made all the time in a LAC-I minus mutant, but also so is lactose permease, the product of the LAC-Y gene. And even one other protein we've yet to mention, an enzyme called lactose transacetylase encoded by the LAC-A gene. All three of these genes are induced by lactose naturally, and somehow that induction involved the LAC-I gene. Now, is there anything odd about these results to you? Mutations that somehow allow these genes to be expressed all the time, but their mutations are not in the genes that are being expressed, but they're in this other gene that we call LAC-I. We're going to come back to that. Now, how does this LAC-I gene work? Does it encode a protein? And if so, what does that LAC-I protein do? How is LAC-I involved in induction of the synthesis of beta-lactamidase, lactose permease, and even this enzyme we don't fully understand its role yet in the biology of E. coli, even to this day, lactose transacetylase. So to get a handle on the role of LAC-I, Monod and Jacob did a very famous experiment along with a postdoc by the name of Arthur Pardee. And it came to be known as the PAJA-MA, really PAJA-MO experiment. The experiment involved the conjugation of a natural E. coli plasmid from one E. coli strain to another. And this natural E. coli plasmid carried an extra copy of the wild-type LAC-I and LAC-Z genes on it. We'll call them LAC-I plus and LAC-Z plus. Now, the recipient cells were LAC-I minus, LAC-Z minus double mutants, so they could never make beta-lactamidase. So they mixed the donor and recipient cells together for a short period of time, and this allowed conjugation to occur. And this plasmid can move from the donor to the recipient and potentially now start making LAC-I and LAC-Z. And then they disrupted the mating pairs. Remember back to like a little blender, you could just kind of vortex them, and that would break those conjugation pili. And then the donor cells were killed by the addition of the antibiotic streptomycin. The recipients were strep resistant. And then at different times after the mating, the cells were broken open and the amount of beta-lactamidase enzyme activity was measured. Now, when lactose was added to the mixture soon after the mating pairs were disrupted, it was observed that beta-lactamidase activity, shown here in the blue dotted line, started up and continued to increase in a direct way over time. All right. However, if no lactose was ever added, no inducer, totally left out of the experiment, they still saw beta-lactamidase activity began to rise soon after the mating, but then it leveled off, as you see in the red solid line. But how do we explain it? How can there be any sort of induction when there's no inducer? Now, the two genes, LAC-I and LAC-Z, come into the recipient cells at roughly the same time. The plasmid is big, but it's not that big. So we can basically assume that conjugation is kind of instantaneous in terms of the two genes. So if the LAC-I encoded protein activity is needed for lactose induction, then we would have both LAC-I and LAC-Z in the same cell. But remember, there's still no inducer around to tell LAC-I to turn on LAC-Z. So how do we explain the minus inducer curve where we see some beta-lactamidase activity for a while and then it levels off? Now, if we add the inducer, lactose, we see beta-lactamidase enzyme activity go up. However, in the absence, we still get some enzyme activity soon after mating. So, question 1. How can any beta-lactamidase be made in the absence of inducer? And question 2, why does the synthesis stop after a short while in the absence of inducer? So the answer comes down to kind of how do we envision the role of this LAC-I gene. If LAC-I, in fact, encodes a protein needed to turn on LAC-Z, then I should never see beta-lactamidase enzyme activity in the absence of inducer. also, since most mutations knock out gene function, if they do anything at all, then a mutation in LAC-I would prevent induction. But remember, just a slide or two ago, a mutation in LAC-I allowed induction all the time. So clearly, LAC-I cannot be a positive player. It can't be required for the induction of LAC-Z and LAC-Y and LAC-A. So what's the alternative explanation? It is basically that the negative of a negative is a positive, to use a mathematical phrase. If mutations in LAC-I if they do anything, knock out LAC-I function, and since that leads to making beta-galactosidase all the time, then somehow the normal presence of LAC-I must inhibit the synthesis of beta-galactosidase. And then induction, when an inducer around, such as lactose, must somehow remove the inhibition. So the negative of a negative, the inducer removing the inhibition by LAC-I, allows the expression of beta-galactosidase. LAC-I is a negative regulator. And in fact, we now call it the LAC repressor. After several more experiments and a lot more theorizing, Minot, Jacob, and Lewolf eventually put forward a model for gene regulation that they called the operon model. Now, by this time, they had figured out that LAC-Z, Y, and A, these three genes that were regulated by LAC-I and turned on by lactose induction, that they, in fact, were right next to each other in the E. coli genome. And they interpreted that as the three genes were sitting adjacent to one another. Now, LAC-I is nearby, but not right next to them. Now, they had to explain how LAC-I encoded a protein that could negatively regulate the synthesis of LAC-Z, Y, and A. And the key intermediate here was the RNA made by transcription. So they hypothesized that these three genes were co-regulated because they, in fact, not only lied next to each other, but they shared a common promoter. And that when RNA was made, all three proteins could be made from that one RNA. And this is what we now call an operon. Now, they also hypothesized that the repressor, LAC-I's protein, acted as a negative regulator that somehow prevented transcription of this operon. So we have two key ideas here. More than one gene sharing a promoter, what we call an operon, and then being co-regulated by a negative regulator, a repressor, that keeps transcription from occurring. These are big ideas, and we'll continue to think about them in part 3 of this video lecture.